just to the previous question, I just highlighted the three essential key limitations that we see in solid tumors, which is the access of T cells to tumor tissue, the heterogeneity of cancer, especially with regards to antigenic expression and to antigenic access, and of course also immune suppression. And so the recently described platform from my team, the so-called PG LALA CAR platform, is a modular platform, a modular cellular platform, which in essence, the concept is that we've built a CAR that targets a unique mutation. It's the PG LALA mutation, which stands for the amino acid exchange that happens at the Fc terminus of a monoclonal antibody. And why we've become interested in that is because several companies, pharmaceutical companies, are engineering these days their monoclonal antibodies for optimized or reduced Fc receptor engagement. And so this PG LALA mutation, what in essence does, it prevents the activation or the engagement by activating Fc receptors. So this has, safety reasons in the end. But in essence, what happens is there are already some antibodies that are on the investigation, that are also under clinical use, but also under preclinical development. So meaning that there is a whole library of already developed monoclonal antibodies that bear this PG LALA mutation and would just target any desirable antigen. And so we felt that this could be a massive advantage in the development of CARs if you just have to develop one type of CAR, so this PG LALA CAR, which would target only this unique mutation not found in patients and not found in humans, with monoclonal antibodies that are really clinical grade, so where they've eventually already undergone some clinical testing, and with the perspective that perhaps this will reduce development time and development costs, because you're just focusing for all of your approaches just on the CAR, and the module is the monoclonal antibody which is already developed. And we've shown that in a number of preclinical in vitro and in vivo models this actually works, that we can target heterogeneous tumors, that we can target multiple antigens, that we can switch the antigen, that could stop giving the antibody, and this would then stop the activity of the CAR. So indicating that this PG LALA CAR could be really a suitable platform. And actually, this PG LALA CAR recently entered clinical trials because this was also a concept developed by our partner Roche, and this, to my knowledge, this has recently entered clinical trials in China.